Not even actually. It's a danger zone over there. But also the wedding was planned. I think I was around the same way.
Good morning. Can you all hear me okay? Okay, thank you. Welcome, Friday. Anything after this session or? Meds, okay. Yeah, review session, right? I'm going to be there. Okay, well, not quite the weekend yet, I guess. Okay, so the topic for today, we're going to talk about continuing in our discussion of enzymology or tangential to our discussion of enzymology. We're going to talk about molecules that aren't proteins but that play a critical role in enzymes that catalyze reactions. So these are colloquially or broadly called cofactors for enzymes, and they many of them are derived from vitamins that are essential in our diets. Okay, so that's going to be kind of the flow of today's discussion. We have a list of different enzyme cofactors, and they all have precursors in the diet with the exception of lipoate. So lipoate is one that, the odd one out there that we can synthesize from simple precursors. But all the other ones have a dietary requirement. So you'll want to focus on the function of those cofactors and what sort of enzymatic activities they support, where that cofactor comes from in the diet, and then what would be the consequences of the loss of that cofactor or the lack of that cofactor. Okay, so that's kind of where you should focus your study. Is that clear? And when I say deficiency, like focus on the lack of that cofactor, that could come from a dietary deficiency. It could also come from inborn errors of metabolism related to the uptake or modification of that cofactor. Okay, so we just, I guess before I get too ahead of myself, why don't we review a little bit from earlier this week? Okay, pretty straightforward, right? If the Km is increasing, that's a competitive inhibitor. And here we have one with the Vmax decreasing. Now, not shown here, but this red, this would have its Vmax and then a half Vmax, and that would line up right with the Km, so the Kms wouldn't change here. And this is defining a non-competitive inhibitor. Great. Okay. Okay, in this case, the fructose 1,6 bisphosphate is decreasing the Km, right? So that means that it's having the same amount of activity but at a lower substrate concentration, so it's a more active enzyme, right? Okay. Okay, so that would fit the definition of an allosteric activator. Okay, so, don't be surprised. You're going to see a question or two on the next exam and on the final exam that may ask you to interpret graphic data or textual data to solve a problem like that. Okay, I'm just going way, way back. to the different levels of protein structure. Okay, good. Quaternary structure, pretty straightforward. Okay, I think I gave you the first page of a clinical scenario. Did anybody have a chance to read that over? Okay, I want you to take a quick moment to read that over. So some of these laboratory values, I think we've talked about hematocrit and the related hemoglobin. So that would basically say how many red blood cells you have. This boy has lower than normal hemoglobin. And if you measured his hematocrit, presumably that would be lower as well. So he has an anemia. The mean corpuscular volume, this is the average volume of each of those red blood cells that he doesn't have enough of. And they're smaller than normal. So this is a microcytic anemia. Make sense? Okay. And then the prothrombin time, this is related to coagulation. And that's basically suggesting that he has a coagulation defect, maybe a platelet disorder. Okay. Any impressions that you'd want to share?
Iron?
Iron? Why would you say iron?
Because of the hemoglobin.
I think that that's reasonable. Yeah, that might be reasonable. I guess it doesn't explain the leg pain. I guess that's kind of doesn't really fit there. Yeah. Scurvy. Okay. What's scurvy? Okay, lack of vitamin C. Okay, so given that hypothesis, what would you do next for this child? Okay, good. I think those are both reasonable. Yeah, good. So we're going to cover this case towards the end of class, and it's pretty interesting, both for as a typical presentation of the nutrient deficit, and also kind of some bigger picture information that I think would help you in your practice, and it would have a big impact on healthcare costs in our country. Okay, should we go on? All right. So what we're talking about today are cofactors, and the basic relationship between a cofactor and an enzyme is depicted here. So an enzyme is a protein. There are RNA enzymes, but let's just put those aside for now. Okay, so an enzyme is a protein, and it's going to have its active site is going to be made of the defined by the amino acid side chains that are making that 3D structure and that chemistry at the active site. So many enzymes are incomplete if you just look at the protein part of it. So many enzymes will require an additional non-protein component that sits within the active site and helps to define that three-dimensional shape and the chemistry that allows it to bind to a substrate. Is that clear? Okay, so to make that just a little bit more detail. So we know from Dr. Cormier's lectures, we get an enzyme. If it's a protein enzyme, it's encoded in DNA. the promoter gets activated and it gets transcribed and then translated on the ribosomes to make a protein. Now, if that protein is an enzyme that requires a cofactor, in the absence of a cofactor, we refer to it as an apoenzyme. So it's an incomplete enzyme. And it's essentially catalytically dead. Okay? So in order to make a holoenzyme, an enzyme that's capable of catalysis, there needs to be that cofactor binding into the active sites and allowing the enzyme to bind its substrate and to catalyze a reaction. Now, lipoic acid being the exception, all the other cofactors that we're talking about today have a precursor vitamin. So these are things that we don't have the ability to synthesize ourselves, so we have to eat them. Okay, so we eat the vitamin precursor. There may be some modification of the molecule by intracellular enzymes to make the final cofactor. Then that cofactor binds to the enzyme to make a holoenzyme, and then that enzyme is competent to bind its substrate and to carry out the chemical reaction. Okay, and just in terms of how that reaction looks in a hypothetical enzyme as shown by Marx, that cofactor is both essential for binding to the substrate and for catalyzing the conversion of the substrate into the product. Is that clear? Okay, so if we look kinetically, this is an example and this, the details of the enzyme aren't important. This is lysine hydroxylase, which uses lysine as one of its substrates, and it's a vitamin C dependent enzyme, but it also uses the flavin adenine dinucleotide as a redox cofactor. So this is showing the at two different concentrations of lysine, one of its substrates, the relationship between the concentration of that cofactor, FAD. And what you can see is that, first of all, there's a relationship between the activity and the substrate concentration, just as you would expect, but it's a really sharp cutoff between the enzyme being essentially off and turned on to Vmax. Okay, so in the absence of the cofactor, the enzyme is basically inactive. Okay, and these black diamonds here, this is showing a single point mutation in that lysine hydroxylase, and that point mutation blocks the binding of FAD, and you can see that even at the highest concentrations of FAD, the capacity to catalyze the reaction is lost, or is greatly compromised. Okay, does that make sense? Okay. All right, so this enzyme, presumably is carrying out an important function within our body, and if you don't have that function, then you're going to have some symptoms or abnormal lab values or what have you that will occur. So the symptoms of vitamin deficiency can occur in a few different ways. So the most straightforward way or easy to understand way is that if you're not eating the vitamin, you're not going to have the cofactor, so you're not going to be making the holoenzyme, so you're not going to be doing this important reaction within our body. Okay. Now that would be referred to as a dietary deficiency of the vitamin. You can also have a functional deficiency of the vitamin, and a functional deficiency of the vitamin would be, this is an example here, say you're eating plenty of the vitamin, you're putting plenty of it into your mouth, but perhaps you have an inborn error of metabolism, a loss of function mutation in an enzyme that modifies that vitamin into the final cofactor. Okay, you would have the same symptoms as the dietary deficiency, but it wouldn't be remedied by just replenishments of the vitamin in the diet. Is that clear? Okay. You can also get symptoms of a dietary deficiency with a loss of function mutation in that that enzyme that is performing that function. Okay, clear? All right. So some cofactors are referred to as prosthetic groups. And these type of cofactors, when the enzyme is synthesized or shortly after it's synthesized, it's going to bind the cofactor in the active site. And in some cases, this is actually a covalent bond between an amino acid side chain and the cofactor. That cofactor is tightly associated with the enzyme for essentially the life of the enzyme. Okay, so it's like, I don't know, a post-translational modification that's almost permanent for the enzyme. Okay, and we've referred to those as prosthetic groups. So just in our hypothetical situation here, it's showing that that cofactor is modified during the reaction. Now, frequently you'll have another reaction that's going to restore that cofactor while it's still associated with the enzyme. And one that we'll discuss, flavonadenine dinucleotide, is one of the more common prosthetic groups. Succinate dehydrogenase in the electron transport chain. This is here we have the hypothetical situation, and then this is a concrete example. Succinate dehydrogenase. The function is to oxidize succinate to fumarate, and while it's doing that, it's transferring electrons from succinate to the redox cofactor. Okay, then in order to regenerate the cofactor, you have to transfer those electrons onto coenzyme Q, and they get transferred down the electron transport chain. Okay, so prosthetic group cofactors such as FAD are distinct because they remain bound to the enzyme. There are other cofactors that bind to the enzyme transiently along with other substrates for the reaction. Okay, so both the cofactor and the substrate bind to the active sites, the reaction occurs, the cofactor then can be restored through another reaction. Okay, but this occurs not while the cofactor is tightly associated with the enzyme. So this is our hypothetical example, and a concrete example is alcohol dehydrogenase. So alcohol dehydrogenase, it's kind of a detoxification / fuel oxidation reaction. So that's going to take the alcohol from ethanol and convert it into an aldehyde, acid aldehyde, and it's going to be transferring electrons from the ethanol to the nicotinamide adenine dinucleotide and reducing it. Okay, so the nicotinamide adenine dinucleotide then, this could be oxidized in several different places as shown here. The lactate dehydrogenase will oxidize it and restore it. Yes?
Example from last week, you mentioned that alcoholics might have higher oxidative stress. Is this why, basically?
No. There is a, so this would explain, and this is kind of getting ahead of ourselves here, but this would explain why an alcoholic might be hypoglycemic in the fasted states, because you get NADH toxicity of the tricarboxylic acid cycle prevents gluconeogenesis from happening. Now, the oxidative stress, that occurs because of NADPH depletion. There's another cytochrome P450 enzyme that can do the same reaction, converting ethanol into acetaldehyd, but that is it's instead of generating NADH, it is depleting NADPH, which is a major source of reducing power for antioxidant defenses. So I think you're going to have some more coverage of ethanol metabolism. It's hugely important in medicine. So, yeah. Okay. Questions? Any other questions? Okay. All right, so this is just a list of different enzyme classes on the left, and then some specific examples of types of enzymes on the right. So the cofactors that we're going to be talking about today are going to be focusing on the redox enzymes and the transferase enzymes. So the redox enzymes transfer electrons from one substrate to another, and the transferase enzymes are transferring carbon or another group from one substrate to another. There is overlap between those two, and the one example that we'll talk about towards the end of class, the pyruvate dehydrogenase complex, that's doing both redox reactions and transfer reactions, so there is overlap. Okay, let's talk about redox reactions and the cofactors that are associated with them first. So redox reactions are the transfer of electrons, and so it's a transfer of electrons, right? But if you're looking at it on paper or in your textbook or on a PowerPoint slide, it's going to be an H, a proton, okay? Because the electrons come with a proton during the transfer. So just kind of anytime you see like H's transfer, that's likely to be a redox reaction. Okay, so with redox reactions, there's always two substrates and always two products. So one of the substrates is going to get reduced and the other is going to get oxidized. Electrons don't just go out into space, they have to go onto another substrate during the redox transfers. Okay, so if you remember this from undergraduate, right? So loss of electrons is oxidation. Gain of electrons is reduction. Okay. So, yeah, I still rely on that today, so it's not always super intuitive. Okay, so some types of redox reactions. There's three kind of broad categories, and again, there's a little bit of overlap between them. So fuel oxidation reactions, so succinate dehydrogenase, in the TCA cycle, that's an obvious example. Alcohol dehydrogenase in ethanol metabolism, you could consider that, both a detoxification reaction. It certainly is, but it also actually generates fuel that can be pretty useful for our body. So fuel oxidation, detoxification, and then biosynthesis. Those are kind of the three broad classes of reactions that these redox cofactors participate in. Okay, did anybody learn the Nernst equation as an undergraduate? Okay, you can forget it. All you have to remember, okay, there's potential energy of the electrons on one substrate, and then it's a different potential energy when it goes to another substrate. Okay, so that is called the redox potential, and you can relate that to the overall energy change in the reaction. Okay, so reactions can be driven forward by redox potential, essentially. Okay, so the redox cofactors that we'll discuss today, the two that are derived from niacin, NADH and NADPH, flavin adenine dinucleotide, and there's also flavin mononucleotide, which should be on here as well. Vitamin C, ascorbic acid, and then metals. So I'll talk a little bit about metal. I think the example I use is iron in the complex 1 of the electron transport chain. In terms of metals, we'll talk about sources in the diet and consequences of nutritional deficiencies, largely in the immunology, hematology, oncology course that you'll take in the spring. So just kind of be aware that they are redox cofactors, but a lot of the nutritional kind of details, just stick a pin in them, and we'll discuss them later. Okay, shall we carry on? All right. So the first redox cofactor that we'll discuss is nicotinamide adenine dinucleotide, and there's a related molecule, nicotinamide adenine dinucleotide phosphate, that comes from is derived from the same vitamin, niacin. So if you look structurally, everything from here down, these are things that we can synthesize with our own enzymes using simple precursors. Okay, so this is the adenine base, the ribose sugar, and then phosphates here. So this is one nucleotide, right? The base, the sugar, and the phosphate. Then this is a ribose sugar with a nitrogen link to another molecule or another part of the molecule. So that's the other nucleotide from the dinucleotide. So this part of the molecule, this is derived from that essential vitamin in our diet, niacin. Okay, we can't synthesize that, but other things that we eat can, so that's where we get it. And that vitamin-derived portion of the NAD, that is where the redox actually happens. So this carbon can either be in the reduced form, that is with electrons packed onto it, or in the oxidized form with electrons stripped away from it. The rest of the molecule, this, if you recall on our discussion of protein structure from a week or so ago, I mentioned that many proteins are evolved for being able to bind adenosine nucleotides, right? It's like a common thing that a lot of proteins can grab onto. So this portion of the molecule, think of that as like the handle, okay? So that's what the substrate binding site of the enzyme is going to be grabbing onto, okay? Because it's a common recognition site. Okay, so this is same molecule from, in a more two-dimensional representation from Marx. So first I'm going to talk about NADH and then NADPH. This is showing both, and the ribose sugar there, that can either be phosphorylated in NADPH or not phosphorylated in NADH. Okay, so let's consider a little bit of detail of the catalysis. So we'll look at lactate dehydrogenase. I think in the earlier reaction, or in the earlier example, I showed this as in the reverse reaction. This is actually a reversible reaction, and within our cells it can go either direction depending on the concentrations of the substrates and the redox potential within the cell. But let's look at this in the case of like a gluconeogenesis situation within the body, for example. Lactate being converted into pyruvate, and then the electrons from lactate being transferred to NAD plus to reduce it to NADH. Okay, so the conversion, the difference between lactate and pyruvate is essentially you're turning an alcohol into a ketone. Okay, so the electrons that are associated with this carbon in the alcohol state go to this carbon on the niacin-derived portion of NADH, and when they do, they reduce it. So this is a 2-electron transfer. You get 2 electrons and 1 proton, and that converts the oxidized NAD plus to the reduced NADH. Okay. Then this reduced form, this can be oxidized on another reaction. For example, the complex 1 of the electron transport chain is NADH oxidase, and that can reverse the reaction, take those electrons off, and restore the oxidized form. Make sense? Okay. Okay, so NADPH, as shown here, the same basic structure, but we just have phosphate hanging off of the ribose sugar, but that phosphate group makes it a quite different molecule, and it participates in different reactions. Now there's glutamate dehydrogenase is an enzyme. It's a redox enzyme that can use either NADPH or NADH as a redox partner. There may be a few others that have, promiscuity for their redox cofactor utilization, but they're rare. Okay, so generally an enzyme is going to use either NADH or NADPH, not both, but there are exceptions. Okay, so while NADH is mainly associated with fuel oxidation reactions and in converting, large substrates into smaller molecules so that we can get energy from them, the NADPH is involved in biosynthetic reactions and in detoxification reactions. Okay, that's the main role there. One of the important classes of enzymes that use NADPH are the cytochrome P450 enzymes. So these enzymes are involved in both detoxification. So if they have a toxic substrate here, for example, denoted by the R, by adding oxygen to that toxic substrate, it makes it more soluble and easier to excrete in the urine. Okay, so it's part of like getting rid of toxic stuff is to make it more and more soluble, right? Also, this addition of oxygen or alcohol groups to substrates, this is important for some biosynthetic reactions as well. So some of the things that we need in our body are made by cytochrome P450 enzymes. And there are examples of cytochrome P450 enzymes that can use NADH, but most of them use NADPH. Okay, so cytochrome P450s, and I think there was a question about ethanol metabolism. That other alternative ethanol metabolism system besides the alcohol dehydrogenase, that's a cytochrome P450 enzyme. Okay, and if you drink a little bit infrequently, all your alcohol goes through alcohol dehydrogenase. If you drink a lot of alcohol frequently, then the cytochrome P450 enzyme gets upregulated and you get a lot of alcohol metabolized through that way. Okay. So another example of NADPH in a biosynthetic role is the synthesis of cholesterol. So the HMG-CoA synthase, we talked about HMG-CoA synthase that makes the beta-hydroxy, beta-methyl-glutarol-CoA a few days ago, I think. So that's one step of the cholesterol synthesis. The rate-limiting step of cholesterol synthesis and the target of the statin classes of drugs is HMG-CoA reductase. And that is converting the beta-hydroxymethylglutarol-CoA into mevalonate. Mevalonate it's got a few other metabolic fates, but mainly it's a substrate for the generation of cholesterol. So that conversion of the HMG to mevalonate, that uses the reducing power of NADPH again. Okay, so just broadly, NADH is involved in fuel oxidation. NADPH is involved in detoxification and biosynthesis. They both require that nicotinamide ring, which is derived from niacin, or also called vitamin B3. Okay, so niacin is essential in our diet. However, in the absence of niacin, in the absence of vitamin B3 in our diet, we can generate some niacin from the amino acid tryptophan. And this is the kynureninase pathway. It requires many enzymes. And just put a pin in it, this pathway is dependent on pyridoxal phosphate, vitamin B6. Okay, so there's some overlap between those two nutrients. Now, if you don't have enough niacin in your diet, generally you can't make enough just from tryptophan. Okay, this pathway is not sufficient to meet your niacin needs. Okay, so where we get niacin in the diet, meat and whole grains are kind of the, where you would get it naturally, I guess. And then many cereals will add a multivitamin essentially into the cereal, so cereals are good sources of vitamins as well. If you don't have sufficient niacin, The symptoms, the triad or quadrad of symptoms, if you will, is referred to as pellagra. So pellagra, I think that means like leathery skin in Italian. And that is referred to, or that refers to the one of the hallmark symptoms of dermatitis or a rash. And that dermatitis, it manifests itself first on sun-exposed areas. So that there's that phenomenon called the Castle's necklace, where you see a rash that kind of encompasses the neck. But you can also see it on other sun-exposed areas on the hands and feet and anywhere else. So pellagra, you would remember it by either the 3 Ds or 4 Ds. So dermatitis, dementia, and diarrhea. Okay, those are the 3 Ds. The 4th D is death. Okay, so this can be fatal if it's not corrected. It can be corrected if the cause is a nutritional deficiency. You can replace niacin, give someone oral niacin supplements, and shown here, this is before treatment, and then after treatment, the dermatitis can largely resolve. Now, in terms of linking the symptoms of the condition to the biochemical functions of NADH and NADPH, there's kind of because NADH and NADPH participate in so many different reactions, there's a lot of different things that contribute to it. So part of it is that NADPH's involvement in antioxidant defenses. There's also a hypothesis that in this kynureninase pathway, this gets upregulated, and there's an intermediate in that kynureninase pathway that that will accumulate, and that can photosensitize skin. Okay. In terms of the diarrhea, diarrhea is caused by rapidly dividing cells in the GI tract, and those are going to be more sensitive to the loss of the NADH and NADPH. Okay. Let's see. Has Dr. Cormier discussed DNA repair yet, or is that coming up? Okay, he will in a few weeks, I think. He may talk about poly-ADP ribosylase, and this is another. Just going to mention this in a couple of medically important contexts as another function of nicotinamide adenine dinucleotide that is independent of its redox form. So there's a class of enzymes called poly-ADP ribosylases, and these all use NAD+ as a substrate, and their catalytic activity is to cut away the nicotinamide ring. And if you cut away the nicotinamide ring, if you cut this off, you have adenosine, ribose, 2 phosphates, and then another ribose. So the poly-ADP ribosylase then transfers the rest of the molecule onto a substrate, and that substrate is a protein, and multiple proteins, including the PARP enzyme itself, can be ribosylated. So this is showing a single ADP ribose binding, and then you get these big branched chains of it. That's where the poly comes in ADP ribosylase. Okay, so the function, the significance of this is twofold in the treatment of cancer and in infectious diseases. In terms of cancer, ADP ribosylation, this is a critical component of DNA repair for many DNA repair pathways. So the poly-ADP ribosylase, this has domains that will recognize and bind sites of DNA damage. When they do that, they become activated and they poly-ADP ribosylate themselves and other substrates. That then recruits different DNA repair enzymes to repair different types of DNA damage. Okay, is that clear? Now, there's a class of drugs called PARP inhibitors that have shown some decent efficacy in anti-cancer treatments. And this is the idea here is you're taking advantage of cells' lack of cancer cells' inability to repair DNA. So it's like the cancer cells have an intrinsic defects in DNA repair mechanisms. So then you're also blocking the PARP1, and that makes them more susceptible to DNA damage, and the cancer cells will die. Okay, but the normal cells don't because they have intact DNA repair systems. So this is it's a synthetic lethal strategy, and it's used in combination with or for treatment of cells that have defects in homologous repair, like with breast cancer, the BRCA1 or BRCA2 negative breast cancers, they would be especially susceptible there. Okay, so ADP ribosylation is important for cancer drugs, and it's also important in infectious diseases, and particularly cholera. So cholera toxin, so kind of shown here, this is the gut with the Vibrio cholera. When the Vibrio cholera bind to the gut epithelial cell, they're going to So the cholera are living out in the lumen of the gut. They're making a cholera toxin that gets endocytosed and internalized into the gut epithelial cells. That cholera toxin has an ADP ribosylation enzymatic activity. So it's going to be consuming NAD in the cell's NAD to ADP ribosylate and constitutively activate protein kinase A. And then when protein kinase A gets activated, you get phosphorylation and activation of the cystic fibrosis transmembrane conductance regulator, the CFTR protein. That then effluxes chloride ions out into the lumen of the gut. And then that causes an osmotic imbalance, so you get a bunch of water flowing into the lumen of the gut, and that causes diarrhea, which will, perpetuate the Vibrio cholera out into the environment. Has Diebel discussed cholera at all? No. Okay, I think he will at some point. All right, so just a couple examples of NAD in a non-redox function. Okay. Questions? Yes.
I guess that we would be expected to that pathway.
No, you should know that it's there. So like this pathway between a G-coupled protein receptor, adenylate cyclase, and protein kinase A, you're going to get that over and over and over and over again. You're going to hear me say protein kinase A probably about 150 times in the next 6 weeks. Okay, so. And you're going to hear it from others as well. So you don't have to know it now. You're going to have to know it eventually. So, okay. Should we move on to the next redox cofactor? Okay. So the next redox cofactor that we'll discuss is the flavin mononucleotide and the flavin adenine dinucleotide. So the vitamin-derived portion is here, this triple ring structure. This then is phosphorylated to become the flavin mononucleotide. Then the phosphorylated form can have the adenosine nucleotide added onto it to make flavin adenine dinucleotide. Okay, so there are two redox cofactors, and they differ by the presence or absence of that adenosine nucleotide. So the flavin mononucleotide and flavin adenine dinucleotide, they are similar to NAD and NADPH in that they take 2 electrons at a time, so they're involved in 2 electron transfers. However, they're different in that it's different places on the molecule that are getting reduced instead of just a single carbon like it is on NAD. Okay, so significant. Not terribly, except that because you have this somewhat stable intermediate, The FMN and FAD, they can do single electron transfers, and that can contribute to oxidative stress and free radical stress. Okay, so this is shown as a one-way reaction here, but this is also reversible, right? So when FAD gets reduced to FAD2H, it can then get oxidized back to FAD. And an example of that, succinate dehydrogenase in the TCA cycle and also in the electron transfer chain. So the kind of one of the common reactions that FAD containing enzymes will participate in is the taking a single carbon-carbon bond and taking electrons away from the carbon and making it into a double bond. Okay, and that's shown here. So the electrons that are shared by these two carbons get transferred to FAD, and then the reduced form of FAD, we refer to this as FAD2H. This then can transfer electrons to coenzyme Q. Coenzyme Q then transfers the electrons ultimately to molecular oxygen to make water in the electron transport chain. Okay, and that's there in the metabolic map if you're interested. Okay, so the vitamin precursor is riboflavin for both FMN and FAD, vitamin B2. So milk, eggs, organ meats like liver, beans, and mushrooms are good sources. So it's pretty widely distributed in the diet. In terms of how the riboflavin vitamin becomes the FMN or FAD cofactor, there is a kinase that will phosphorylate the riboflavin to make flavin mononucleotide. Then the FAD synthetase will add that adenosine nucleotide to make the flavin adenine dinucleotide. So biosynthesis is pretty simple. Okay, so the riboflavin deficiency, this can have some constitutional symptoms and just like fatigue. I think I suffered from that just momentarily. Fatigue and opportunistic infections and such. There's a couple of characteristics that occur with the vitamin B2 deficiency, the chelosis or the sores on the sides of the mouth, and then glossitis, which is sometimes referred to as a beefy tongue. So it's your tongue gets swollen and can be painful. So those are associated with vitamin B2 deficiency. Now, in order to diagnose vitamin B2 deficiency, or really to diagnose any vitamin deficiency, you would be looking at the presenting symptoms, inquiring about the diet, certainly, like knowing what they eat and what they don't. And then a final strategy, a biochemical strategy, is to, if you suspect one vitamin is deficient, you pull an enzyme that requires that vitamin out of the patient and then interrogate its activity. Does that make sense? Okay, so I'll illustrate this with the diagnosis of the riboflavin deficiency using the erythrocyte glutathione reductase assay, but this strategy can be applied to other vitamin deficiencies as well. Okay, so glutathione reductase, this is an enzyme that's present in red blood cells and in many other cells. But red blood cells, that's an easy sample to get from the patient, right? So you can get the red blood cell glutathione reductase pretty easily. Okay, so the glutathione reductase, this is an enzyme that uses flavin adenine dinucleotide as a prosthetic group cofactor. And the enzymatic function of glutathione reductase is right in its name. Okay, it's taking electrons from NADPH and transferring them through FAD, and then using those electrons to reduce the oxidized bond between sulfur atoms on oxidized glutathione. Okay, so glutathione is a tripeptide, and it's really important for antioxidant defenses and some detoxification reactions. One of the amino acids in glutathione is a cysteine, and it has this free sulfur on it. That sulfur, shown here, that's reduced, and then it can be oxidized by joining to another molecule of glutathione through that sulfur to make a disulfide bond. Okay, so glutathione, it's kind of like a redox cofactor in a sense. And generally within the cell, we want to have a lot of the reduced glutathione because the reduced glutathione is useful for detoxifying hydrogen peroxide, for example, and for adding on to things that we want to get rid of to excrete. Okay, it's 10:50. Maybe I should stop here and catch my breath and resume our discussion at 11 o'clock. Okay.
Just a heart. No, it's nice to have reconfirm.